Let's try this one. So number 2 on Punnett square practice number 1. All right. So first we're gonna define our traits here, our variables. I'm sorry. And this guy right here is big R, little r. So I write it right here: big R, little r. Big R equals red. See the red? And little r is white. Now we're going to write all combinations of those variables, so it's always the same. Homozygous dominant, homozygous recessive, heterozygous. Hetero means different, homozygous means same, and then we know it's dominant because it's the dominant trait. The capital R is the dominant. Heterozygous recessive, I'm sorry, homozygous recessive, I messed up there, right? And heterozygous. So now we're going to put the phenotype for each genotype. This is the genotype, the letters, you can't see them. The phenotype is a physical trait you can see. The phenotype for big R would be red. 2 reds is going to give you red. The phenotype for a big capital R, small r is, let me add that letter there, is red as well, because the red is dominant to the white because it's capital letter. There's no blending, there's no pink. And little r, lowercase r, we have white. So we are doing complete dominance genetics, called Mendelian genetics. There is something called incomplete where you would get a blend. We're not doing that. This is red, red, and white. There's no blending. So what's the cross? So we go back up to the question. It's asking for a homozygous dominant red plant with a heterozygous red plant. Homozygous dominant, I know, is this one. And it's red, so it works out. And it's heterozygous, I know, is this guy. So we're going to cross these two. So I'm putting the cross in, and I'm going to do the red in red. That makes sense. So this is going to be our red. Capital R. And when you do this, you always want to write the cross above where you're going to do it. And then we're going to do the white. Can't write in white, so we'll do the white in gray. This is going to be gray. Heterozygous, big R, small r. So first we're going to distribute the heterozygote. It's going to be on top. Remember, I always drop the one on the left to the side and the one on the right here on top. So I'm putting the big R here, small r here, and now they are totally separate, right? And we're going to put the going to drop these down here, capital R, and they are separate. We're going to distribute the letters into the box now. This is called the Punnett square, right? So I have a big R here, a capital R. I'm going to put it right there. Capital R, I'm going to drop it here. We're going to put one into each box. And the little r, we're going to distribute going down. So R goes in here and here. Oops. See? They can get tricky, right? There you go. And we'll do the same thing here. R going across. Now we have to write in the box what each one is. So we'll write the red ones, and if it has a capital, even one capital R, it's red. So I see red here. I'm writing in the box red. I see a capital R, little r, but that's still red. You look at our table, it's red. Capital R, red. And capital R, little r, red. Hopefully you make your boxes a little bigger so you can write in them. But if you write the phenotype in the box, you won't get confused later when the questions, because the questions can be a little more complicated in this. It's asking each chance of expression, the dominant trait. Dominant is red. We could just write that above there. Dominant trait. And the recessive trait is white for this plant. And we have that right here. We know that. So how many are red? We count. 1, 2, 3, 4. 4 of the 4. There's only 4, so 4 out of 4 red. And 0 out of 4 white. So let's write it over here. 100%, 4 out of 4. Red. Always add the phenotype at the end so we know what you're talking about. And 0, white. Some people would just do this. They'll just put 100%, 100% and 0%. You did all this work and you figured it out, but I cannot read it and know what you're talking about. You have to add the red. I know this went off, but white. You have to label it.